basically where we left off was talking about chromosome changes. And so with chromosome changes, it's basically just within the chromosome, something is different than it should be. Because in chromosome structure, we need our chromosomes to be, or we need our genes to be on the same chromosome, we need them to be in the right order, we need the right spaces in between, all that stuff for them to be used correctly. And we talked about deletions, we talked about duplications, and so now we're kind of getting into a little bit more detail about these inversions. And the problem with inversions, and if you recall, an inversion is where you have, so this is the normal chromosome, and what happens is you have two breaks, again, just like with your deletions that are inside, but instead of losing that piece, what happens is the piece kind of flips around, and there are different ways that that happens, but the result is that it flips around so that you can have a different order in the chromosome. So if these represent genes, so gene A, gene B, gene C, etc., if you look at the break point between C and D and between G and H, you're just flipping that piece around. So you just have this backwards piece. Same thing over here. We've inverted between A and B and between the centromere and gene E. And what that does is that it just flips those genes around. They're just going the opposite direction. And if you kind of remember, the direction of the gene has to do with, is it on the top strand or the bottom strand? And so now you just put it on a different strand. So you just flipped it upside down. Now depending on whether it involves the chromosome, the centromere, or whether it doesn't involve the centromere, has a lot to do with the problems that it causes. And so if it's around the centromere, so it includes turning the centromere upside down, it's called a pericentric, like perimeter, so it's around the centromere. But if it's on one side, it's a para. So they sound nice and similar to each other, so it's kind of hard to remember which one is which. But if you just remember like the word perimeter, that just tells you that it's around the centromere. So all we're talking about is the order that's getting changed. We're not deleting, we're not duplicating, we're not doing anything else, but just flipping it upside down. Now, usually it has no effect on the individual that this occurs in. Usually because you're not losing information, you're not changing it. There may be some slight differences in how the genes are used, but for the most part, these individuals are okay. Occasionally, you might have something called a breakpoint effect, which may mean that instead of breaking between genes, maybe it breaks in the middle of a gene, and then that would be a breakpoint effect because you've basically broken your gene in half. And so now that gene is kind of destroyed. And you can also have something called a positional effect, because if you are breaking, so here our break was between C and D, if that's a really big region that has a lot of control parts in it, like controlling genes or controlling chromatin, etc., like maybe it's an imprinted region, if you break it there and move it into a non-imprinted region, that would be a positional effect. So you've moved it into a region that now behaves differently. Do you remember what imprinting was? Where it's turned off in one parent and turned on in the other. So if you move it into a region where it's not supposed to be on or off, then you might have some positional effects. But usually you don't have a phenotype at all. The problem comes in the next generation, as I kind of implied on, let's say Friday, as I implied on Wednesday. What happens is during meiosis, your crossing over doesn't work quite well. So here we have our pericentric, which is on the two sides of the centromere. Here we have our paracentric, which is on one side of the centromere. Now when they pair up as homologs in meiosis to do all that nice crossing over, where the breaks are has a lot to do with the event eventual outcomes of the sister chromatids that are actually crossed over. Right? So crossing over occurs in meiosis. So where the crossover occurs has a lot to do with what those chromosomes are going to look like when they come out of meiosis. So just like with any crossing over, you wind up with your two parent chromosomes, which are exactly the same as the parent, because we're only looking at the ones that are in the inside that are actually doing the crossing over. So if you look at these two, and you look at these two, we have different outcomes. Now, in both of them, the way that the tetrad forms, or the way that the homologs pair together, is unusual. And that's because they're trying to pair up gene for gene. And so here, where we have our homologs, we have our all dominant at the top. Just obviously, we aren't always going to have an all dominant chromosome. We have an all dominant up there and an all recessive here, so that you can kind of follow what's going on. But if you have gene D, and gene D was actually inverted to the other side of the chromosome, well, this chromosome is going to have to twist around to get the two Ds together. And so it creates something that's called an inversion loop in that meiosis crossing over process. So this is only a problem when you go into meiosis. So it only becomes a problem when you reproduce. Now the outcome of that, if you follow the chromosomes and you follow the crossover that happens between the two sister chromatids, this is going to be big A, big B, big C, big D, big E, all the way until you get to the crossing over spot, and then it's going to cross over to the blue chromosome, which is little f, little g, little c, little b, little a. So what you've done is you've created a duplication of part of the chromosome. So you wind up with one chromosome that has a duplicated part on it. And the other chromosome is going to have a deleted part because that was supposed to be here, but it's not there due to the crossover. And so you wind up with unbalanced chromosomes that come out of it. Now if you add this to a normal gamete, so if this is a gamete and you add it to a normal gamete, you're going to wind up with 3 copies of these A, B, and C genes. And so that can be a problem in that offspring. Same thing with this. If you add this to a normal chromosome, then you're going to wind up missing 1 copy of the A, B, and C, and that individual will just have the 1 chromosome. So it's just like it had a deletion, and their phenotype will show these deletions. Now with the paracentric, what happens is a little bit more interesting. We also have the 2 parent chromosomes. But what happens in these crossover sister chromatids, you wind up with not just unbalanced chromosomes, but unbalanced centromeres. So one of them winds up with no centromere at all, and the other one winds up with both. And so if you put this chromosome and that chromosome into meiosis 2, what would happen to that one? There's no centromere. How can it be separated? It can't be separated because it has no centromere to attach to the spindle. So you're going to lose that. So you're going to wind up with a gamete that has no chromosome in it. And this one, if you have 2 centromeres, what's going to happen to this one? It's going to attach both ways. And so it's going to get ripped apart when meiosis 2 is trying to separate those centromeres. So you wind up with two kinds of gametes that have no chromosome in them, which means they probably won't survive reproduction. So the only outcome you'll get are these normal individuals, but only half the time will you actually have fertility. You see, it decreases fertility because half the time you're working with gametes that will never produce an offspring. Now the problem with this is that these two are going to produce offspring. One that has a duplication, which probably won't be a problem, but the other that's going to have a deletion that is going to be a problem, definitely, because they're missing those genes. So out of the two types, they both decrease fertility, but this one is not going to produce abnormal offspring. This one is. Everybody see that? So if you have an inversion in yourself, it's not a big deal in you, but if you try to pass it down to your children, depending on which type of inversion you have, you may have 100% success at having healthy kids, but 50% fertility. Or you may have 100% fertility, but 50% unhealthy kids. Right? So you can imagine what that would be like having an inverted chromosome. Okay? And women who have multiple miscarriages may actually have some of these inversions that are causing these problems, that are causing these infertility issues with chromosomes. That's why a lot of times they do the karyotype of the tissue that's left over, the fetal tissue. Now translocations are a little bit harder to think about because, first of all, because the pictures aren't great. It's kind of hard to follow because you're kind of trying to see a process, but you're seeing pictures. So here we have an example of two different chromosomes. And we know they're different because they have different shapes. So in this case, we have a breakage of the chromosome down here, but this one's fine. But when the cell tries to fix that, instead of attaching it back to its own chromosome, it just finds a chromosome end that it can stick it on, and it sticks it on the end of this chromosome. So what you wind up with is a new chromosome called a translocated chromosome that has bits of that chromosome attached to itself. And so you get this kind of brand new combination. Now that brand new combination may be beneficial, or it may be neutral, or it may be detrimental. We don't know. It just depends on what chromosomes and genes we're talking about. But when this individual goes into meiosis, it's going to create a problem in pairing again, because what does this chromosome actually pair with? It can pair with this other one, its actual homolog, or it can pair with this little bitty bit that comes from another chromosome. And so you get these weird little structures. Now, if you have these chromosomes in your cell, so you have one from one homolog, one from the other homolog, and then your other chromosome is basically just this kind of mishmash of two chromosomes, and this is kind of your normal chromosome complement. Well, when you go into meiosis, you may have two normal chromosomes, one from each of these. You may wind up with the translocated one and then the broken one, which would be one of these and one of these. Or you may wind up with the translocated one and a full pink one. Or you may wind up with a broken gray one and a full gray one. So you wind up with these different combinations of either missing or additional chromosomal material, depending on what translocation you have and what you actually get from your gametes. Now, when we start looking at some translocations, some are more common than others. And this is an example of a bunch of different translocations between chromosomes 9 and chromosome 22. And the way you would write that is you would say, we're dealing with chromosome 9, and then we also have a translocated chromosome 9 that has part of 22, and And from chromosome 9, it broke at Q34, and chromosome 22, it broke at Q11. And then this is your normal 22. So you have a different way of writing up what these problems are, what this translocation is. Now, in this case, we have our normal, this is a 222. This is a 922. And here we have chromosome 9, which is, medium size. We have chromosome 22, which is tiny. And we see this breakpoint a lot where we break off the bottom of chromosome 22 and we add it to the bottom of chromosome 9. And so if you can see this dark band here from 22, it's this dark band that's been added to the second chromosome 9. And we have it missing here because we have lost that dark band from the normal 22 and it is missing from here. Now, if you have these 4 chromosomes, it's balanced, which means you haven't lost anything, you haven't added anything, and typically that would be an okay condition. But if you have this specific derivative chromosome, it creates an oncogene. You know what an oncogene is?
You've heard of them before. Does it have to do with cancer?
It causes cancer. So an oncogene has been mutated to become a cancer-causing protein. And so by making this particular change, it has created an oncogene where there wasn't one before. And that's actually quite common in a pediatric form of leukemia. So often to diagnose that particular leukemia, they will look for this particular translocation. Okay? So it's not something you're inherited with, it's something that you acquire in your lifetime. Now sometimes we have an unusual type of translocation that happens, and that's where you take your acrocentric chromosomes, remember the ones where the centromere is kind of towards the top? And so your acrocentric ones are 13, 14, 15, 21, and 22. So those are the 5 of those. And in us, in humans, that is also where our ribosomal genes are. So all of the genes that are involved in making the ribosomal RNA are going to be found on those 5 chromosomes. Now, for some reason, it also creates something called a microsatellite or fragile site or something like that at the very tip of those chromosomes. And so they break off easily. And when they break off, they can attach to other chromosomes just like them that have also broken off. And so what happens is you basically break the centromeres in half, and then you stick them back together, but you're sticking the wrong ends together. And that is a special type of translocation called a Robertsonian translocation. So it's named after Dr. Robertson. And so when you have a chromosome that's, say, 1321, it would be 13 and 21 stuck together. Okay. And so what happens is you wind up with kind of an unbalanced, you may have your normal 2 chromosomes, but you can have your Robertsonian translocation kind of has the same thing. The problem there is that what's broken off and lost are some ribosomal genes. So these translocations are often kind of associated with problems in making ribosomes, right? Because we use and this is really cool, we use different ribosomal genes in different stages of development. So we have different ribosomes early on than we do later on in our lives, which is really cool.
They produce different proteins?
Slightly different RNAs, yep. The genes are slightly different. That's just mechanism. All right. So when you have, all right, so if we have our individual who has the translocation, they have 2 all there. It's just in a different place. No effects, no phenotype, nothing associated with it. But when they go into meiosis, what we wind up with in crossing over is instead of just having the homologous pairs independent of each other, now we have this overlap where that pair is attracted to this pair because of this little bitty gray bit here. And so this gray bit is going to be attracted to this homologous area on the gray chromosome. And so instead of having just the pair of homologs, you wind up with 4 chromosomes together. And so crossing over can happen between these two in the middle, between these two in the middle, or between these two. So there are multiple ways that crossovers can occur, and depending on how they're pulled back apart after crossing over, gives us different ways of having the information. So if we have crossovers between these two, because they're attached, you may actually cross over between 2 different chromosomes. These aren't different chromatids. These aren't sister chromatids. These aren't homologs. These are completely different chromosomes. And you can actually compound the problem because you're starting to cross over with the wrong thing. And that can cause a lot of different problems in rearranging multiple chromosomes together. Now, if everything goes right, and you do crossing over the right way between homologs, you wind up with different types of combinations of how they're going to separate in meiosis 2. Now, a normal gamete would have 1 of each, and so if you're adding a normal gamete to our translocation gametes, from this arrangement you can get 1 normal of that and 1 normal of that. So that's what this would be. You can get 1 translocated of that. You can get a deleted one of that, and that would be balanced. But you can also get 1 of these translocated with a full 1 of these, and that would be this one where you get a little bit of extra. Because if you add it to a normal gamete, you get 1, 2, 3 copies of that region. Or you might get 1 of these normal ones and 1 of these deleted ones, which means that you would have a deletion from that end. Okay? So out of all the possibilities, you can have completely normal, you can have a carrier of the translocation, you can have a duplication, or you can have a deletion. Okay? So these 2 are probably going to cause phenotypes because you have extra stuff or missing stuff, but the other 2 will be pretty normal. And this one will actually be just like their parent. Whatever their phenotype is, they'll have the same one. All right? So a lot of these issues with chromosomes aren't really as harmful to the individual as they are to the offspring of the individual. Okay. They usually reduce fertility. They usually cause issues in that way. Now, one type of Robertsonian translocation that's pretty common is one that goes between 14 and 21. And that's a really common one because if you think about chromosome 21 is tiny. It's the smallest chromosome we have. And so if you have a Robertsonian of 14 and 21, then that means that you have basically all of 14 except for your ribosomal genes doubled, right? So it's most of 14, 1 copy of ribosomal genes, you're missing that copy of ribosomal genes. Your other 21, you have the full Q arm. Here we have the full Q arm also attached. And then we have the ribosomal genes here. We're missing the ribosomal genes from this one, okay? But overall, this one is balanced. Now, if that individual goes through gamete formation, the way that it pairs up, it has to pick who it's going to pair with, right? Because we're talking about pairing homologs. So we can pair these two together. We can pair these two together. But this one cannot go with both. Does that make sense? Because it's only one chromosome, it can pair with this one or it can pair with that one, but not both. And so when you start looking at the gametes that are produced, you wind up with a lot of unequal gametes. So you have some that are missing chromosome 21, you have some that are missing chromosome 14, you have some that have extra chromosome 21, and you have some that have extra 14, depending on how they arrange in meiosis. So if you're lucky, you get one of each. You get the 2 parental chromosomes. If you're also lucky, you'll get just the balanced translocation. So you only get 1 chromosome out of that. You might wind up with the translocation and one of the chromosomes. You may wind up with just 1 chromosome, or the translocation and the other chromosome, depending how they sort out. Now, if you add those to a normal gamete that has 1 of each, then you can kind of see how you'd have problems. So with your normal gametes, you're going to be nice and balanced. You'll have 1 of each. You get 1 blue from that parent, a blue from that parent, you get a pink from that parent, you get a pink from that parent, you're nice. It's supposed to be, that's exactly what it's supposed to be, diploid. Now if you have this one, and you add it to this individual, 1 full blue, 1 mostly full blue here, and then you get the full pink and the mostly full pink over here, and you wind up pretty much balanced. Basically the same as the parent. But they still carry the translocation. Now, if you add it to this individual that already has 2 copies of 21, you add it to a 3rd copy of 21, you wind up with an inherited form of trisomy 21, which is also known as Down syndrome. So this is how you get Down syndrome passed down in families. Someone in the family had this translocation, and then they just were kind of unfortunate in passing down both copies. Now these 2, because they're missing the other chromosome, this one would be missing 21, this one would be missing 14, and so those don't work. And then this one over here would be too many copies of 14, which is not consistent with life, right? We don't see these individuals living, okay? So the only outcomes you would get a normal individual, you would get someone who's carrying that translocation and can pass it down again, or you would get someone who has Down syndrome. So you really only have 3 possibilities of phenotypes coming out of an individual who has one of these types of translocations, right? Now, what is trisomy 21? So trisomy 21 is having 3 copies of chromosome 21. This is the most common type of syndrome in humans where you have 3 copies of an entire chromosome. Now with familial Down syndrome, obviously we wind up with our translocation. And here we have our translocation. It's a little bit hard to see, but that little bit at the end of that chromosome is actually one of these. A little bit hard to see. But that's an individual who carries Down syndrome in their chromosomes. It's not, usually Down syndrome is kind of a product of aging of the parents. Older parents tend to have problems with their chromosomes segregating more often than younger parents. Now an example of what's going on when you have translocations. What happens is it creates a structure in meiosis that's called a holiday structure, after Dr. Holliday, not the one in Back to the Future. Or no, Doc Holliday was in, what was that movie, that Western movie? I don't even remember, Young Guns or something. So here we have our purple homologs, we have our blue homologs, but where we have crossovers, we have the structure where this part of the blue is binding with this part of the blue, this part of the purple is binding with that part of the purple, the rest of the purple is binding with that part of the purple, and the rest of the blue is binding with that part of the blue. And so you wind up with these weird little structures. Someone earned a Nobel Prize resolving these structures, figuring out how they actually divide. Because when they do divide, they can divide this way, they can divide that way, they can divide that way, or they can divide that way. They're really cool structures. And that's what all of this is all about, is the different ways that they can divide and produce offspring. But we're not going to go into that detail because I don't want you to have to memorize it. All right, so kind of the overall message. Inversions and translocations both rely on what uncommon occurrences? Which one of those is uncommon? Double chromosome breaks. So chromosome breaks aren't that uncommon, but to get two on the same chromosome is unusual. And so if you get two on the same chromosome, oftentimes your cell is not able to deal with both of them. It can deal with one, but not the other. And so two chromosome breaks are usually what leads to these inversions. It leads to those interstitial deletions. It leads to our translocations because you have to have breakages on two different chromosomes at the same time. And that is why cancer is related to high radiation doses, right? You go to your dentist and they ask you how many x-rays have you had this year? They should. If they don't. Because that x-ray radiation can damage your DNA. Now, your body can take care of a small dose. It can repair all those chromosome breaks. But if you have a lot of radiation happening, like if you were near Chernobyl when it blew up, do you guys know what I'm referring to? I think the Netflix series came out about Chernobyl. Is that? Anyway, that was big when I was in high school. If you were near a big radioactive blast, then all of the DNA in your body would be broken at the same time, and your body would not be able to repair all of it. So you would have more and more translocations happening, and that's why you're getting more and more cancers if you're exposed to a lot of radiation. Also, your offspring would have more problems. You would have more translocations in your family, etc. So it's kind of rare to have both of those breaks occurring in the same chromosome at the same time. All right, we kind of already answered that question. And then the last bit just has to do with some semantics, and it's really just definitions. So the last bit has to do with how many full chromosomes. So we looked at changes in like structure of chromosomes, but this has to do more with the number of chromosomes. So when I say ploidy, what term does that make you think of? Haploid, diploid, right. So that's exactly what we're talking about. And what does diploid mean? Two of every chromosome. It has to do with the count of the chromosomes. So the ploidy has to do with the count of the chromosomes, but a ploid is a set, is a full set. So diploid would be two full sets of chromosomes. So when we're talking about ploidy, there are two ways to think of ploidy. You can think of it as true sets, or euploid, meaning that you have, if you're 2N, you have 2 full sets of chromosomes. If you're haploid, you have 1 full set of chromosomes, or N, right? We use the letter N. So when you talk about euploidy, the ploidy would be the number of sets you have. So we know that we have haploid gametes, and we know that most of our cells are diploid. But some organisms have triploid cells, some organisms have tetraploid cells, some organisms even have octaploid cells, which means they have 8 copies of every chromosome. But the important thing is that they're full sets. So you've got all the way from chromosome 1 to chromosome 22 represented times the number, right? Times the number of sets. Now, the term aneuploidy, so an means not, so aneuploidy would be not a full set. So this refers to when you're missing a chromosome or you have an additional chromosome in your set. So you can be 2N plus 1, which would be aneuploid, meaning that you have 2 full sets, but then you have an extra chromosome. So that gives us pretty much you have either in your set you have a monosomy, which means you only have 1 chromosome in your pair if you're supposed to be diploid. Trisomy means you have 3 chromosomes when you're supposed to be diploid. And tetrasomy means you have 4 chromosomes when you're supposed to be diploid, right? 4 instead of 2. And that refers to individual chromosome counts. So this would be individual chromosome counts. This would be sets of counts of sets of chromosomes. So when we think about aneuploidy, and that's what we typically see in humans, because we can't be tetraploid, that just doesn't work for us. Although we do have cells in our body that are dodecaploid, which means you have 12 copies. Our liver cells are notoriously dodecaploid. Here we have an individual that's normal, diploid individual, 2 of every chromosome. Here we have an aneuploid individual, 2 of every chromosome plus 1. Here we have an aneuploid individual, 2 of every chromosome minus 1. Right? So we would refer to this grouping as a trisomy of whatever chromosome that is. So in this case, this is chromosome 2. So it's trisomy 2. Over here, we only have 1, so we refer to that chromosome set as monosomy 2. This is chromosome 2. All right. Now, this usually creates an abnormal condition. If you have a trisomy, that means you have a full extra chromosome, which means that your protein production is going to be about 150% of normal. And it's not for 1 gene, it's for all of the genes, and so that typically creates a phenotype. Same thing for monosomies. Instead of having your genotype of 2 alleles, you wind up with only half the protein produced. It's kind of like being hemizygous kind of thing. You only have 1 copy, okay? Now, how is that actually caused? It happens during meiosis. So when you think about meiosis, the purpose of meiosis is to create haploid cells. And by creating haploid cells, basically what we're doing is we're splitting the homologs, and then we're splitting the sister chromatins. But if that doesn't happen correctly, then you can wind up with gametes that have extra chromosomes or they're missing chromosomes, and that's how you wind up with an individual who either has trisomy or monosomy. And you can actually track which division, whether it was meiosis 1 or meiosis 2, where the problem actually happened. So here we have our non-disjunction in meiosis 1, so we have our homolog pairs that are being separated. So if we're separating them in the first meiosis, if these separate correctly, but these do not, they both go to the same side, that means that in meiosis 2, you're going to separate your sister chromatids and you're going to wind up with two of the same chromosome in those gametes. And over here, we're going to wind up with none of those because they all went to the other side in that first division. So we wind up with N plus 1 gametes in minus 1 gametes. It's kind of half and half. All of the gametes are wrong. Now if we have the problem in meiosis 2, we might wind up with some normal gametes because at the beginning we have the proper segregation, so we wind up with 2 and 2, but in the second meiosis, we have both sister chromatids going to one side, so only the one gamete has an extra chromosome. The other side from this one is missing one. But because these were okay from that first division, these two are fine. They are full euploid sets of chromosomes. Okay. So depending on the parent, this is actually related to age of either the mother or the father. And that has to do with the fact that when an egg goes into meiosis, does anybody know when an egg goes into meiosis? When it starts? Well, it finishes after fertilization, absolutely. But the start of meiosis happens when your gonads are formed. So when you're a fetus developing in your mother's uterus is when your eggs begin meiosis. So they've been in meiosis since maybe the 12th week of pregnancy, your 12th week, right? And they continue in meiosis until menses each month. That's when it kind of keeps going, and then it finishes after fertilization. So think about how long it takes for meiosis to complete in a human. That can be 20 years, 30 years, right? So depending on how long you wait to have your kids, the later you wait, the longer those cells have been sitting there in meiosis, and they're really kind of suspended right in the middle. And so you have this age phenotype along with it. But there's also an age phenotype. The younger you are, the more likely this is to happen as well. Now with fathers, we don't really see an age effect until fathers become about 70 years old, which is, to me, is just like, whoa! Y'all know who Larry King is? He famously has kids and he's like 90. They had babies at 80. I don't know, it's just weird. Thinking that men's fertility lasts forever and women's fertility stops at a certain point. Occasionally you can have mitotic non-disjunction, and that's just when mitosis fails to separate sister chromatids in a normal cell division. This can have impacts on organisms if their cells are kind of missing chromosomes. The phenotype isn't going to be in the entire organism, it's just going to be in the cells that are affected, that are in the lineage of that one cell that actually failed to separate correctly. And then this is just the graph of the probability of a non-disjunction at age 50 is 1 out of 12 conceptions, although there aren't a whole lot of 50-year-olds who are conceiving, so the data kind of is small at this end of the chart. But the optimal time to have children is in your 20s according to biology, not according to society, but according to biology. And so as you get older, the gap between 35 and 40 is when the testing begins. So they'll start testing you for these non-disjunction phenotypes. I have a few slides that talk about these some of the non-disjunction phenotypes. Trisomy 21 we've already mentioned. We have 3 21s. That has a very specific phenotype associated with the genes that are on that chromosome. A lot of the genes on 21 are aging-related genes, And so kids who have Down syndrome, because they have extra copies, they tend to age faster. And that's why they have a lower life expectancy, because they typically become symptomatic with age-related diseases. So in their 30s and 40s. So their lifespan is usually reduced by about 20 years. Trisomy 13, you have 3 of chromosome 13. That's an unusual syndrome because 13 is much bigger, but there are fewer critical genes on that chromosome, so you can actually find these individuals. And then Edwards syndrome, where you have 3 18s over here. That one is a little bit harder to, or the survival rate is a little bit lower. Usually these guys don't live past a couple of years. There have been some trisomy 13 kids that have lived into their teens. So very severe problems with brain development there. All right. So chromosome aneuploidy involves a change in the number of a certain chromosome in a set. So if you're talking about a 2N diploid individual, which of the following would tell us that it is an aneuploidy? C. Good spot. That was quick. Right? Because aneuploidies have to do with not full sets. These are all full sets. So when you see ploid, that tells you it's a full set. When you see somy, that refers to a chromosome, that tells you that it's a aneuploidy. Right? So that kind of helps you with the terms. All right, and I think that's all. Yeah. Just a little mention about ploidies. Not a important thing, but many of our crops are actually polyploid. Bananas, depending on the size of the banana, so the smaller bananas have fewer sets of chromosomes. Those giant bananas that you sometimes get in the grocery store might have 10 or 12 copies. So the size of the fruit is directly correlated with how many ploids that fruit actually has, which is kind of cool. And that's why they're sterile. That's why you can't plant a seed from a grocery store fruit and expect it to produce fruit, because they are sterile. All right, I'm stopping there. Questions? Thank you for showing up on the Friday before fall break.
Yay! So how long is fall break?
2 days. Just Monday and Tuesday. Yeah. And I hope you all have the opportunity to take that time off. Not everyone will.
I'm not.
Have a good long weekend, everyone.
Big man. I'm going home.
And if you want to join the biology club, just send me a message. I'll add you to the group. You were added, I think. Yeah. We met. I added you to the Microsoft group.
Does it show it here?
It should be in your inbox somewhere.
Can I be added?
I think you were you in the group me? No, okay. Yeah, I'll have to add you to the group. Because, stop.